In the past few videos, what we focused on are a couple of classes of biomolecules that are important in the field of chemical biology. Namely, we focused on proteins, which are constructed from amino acids as their monomeric units. And we then focused on carbohydrates, which are constructed from monosaccharides as the individual sugar units that compose those complex carbohydrate molecules. Now what we're going to do is, continuing this journey, we are going to look in this unit at lipids. So as we look at lipids, I'm going to go ahead and get the whiteboard going here so that we can look at what points about lipids are relevant to our study of chemical biology this semester. So as we look at lipids, much like when we think about how, as we evaluated carbohydrates and proteins, that we had monomeric units that would assemble together to form more complex molecules by the joining of the individual units via covalent bonds. Lipids, similar. So with proteins, we had amino acids joining together to form these peptide chains. With the carbohydrates, we had monosaccharides linking together to form the complex carbohydrate molecules. With lipids, what we have will be a type of molecule referred to as a monomeric lipid. And what we will see is that monomeric lipids will link together through a variety of different types of covalent bonds and other modifications in order to yield complex lipids. And generally what we're going to categorize as the complex lipids are structures that have undergone chemical modifications, such as phosphorylation, so they have phosphate groups. An example of this would be the phospholipid bilayer. That's the bilayer that forms the cell membrane, is heavy in lipid components, the phospholipid bilayer of the cell. Additionally, we would observe that molecules such as steroids represent complex lipids. And additionally, there are a variety of chemical signals within cells that fall into this category of complex lipids. And what happens in forming a chemical signal is that the monomeric simple lipids are condensed together to form longer chains that cyclize or form unique structures or have unique enzyme catalyzed modifications occur to create really specific, highly tailored molecules that travel about the body to send a signal from one portion of the body to another to indicate some sort of change needs to take place in the body. So those are a few examples of the complex lipids that we can think about. Those complex lipids are constructed from monomeric lipids by either joining multiple simple lipid units together and by completing structural modifications of those simple lipids. When we talk about a monomeric lipid, that being a molecular weight of about 100 to 1,000 Daltons, with a major portion of that is going to be comprised of an aliphatic or aromatic hydrocarbon. So when we talk about monomeric lipids, we're looking at molecules that have a molecular weight of about 100 to 1,000 Daltons. And the major part of that molecule is going to be composed of an aliphatic, meaning fatty, meaning hydrocarbon, or aromatic, specifically having a 6-atom ring with that alternating pattern of double and single bonds, is going to be what we will see here. The main example type of monomeric lipids that we need to be aware of are fatty acids, which are the building blocks for a variety of complex lipids, such as the steroid molecules are composed of fatty acids. Fatty acids, being monomeric lipids, are produced by a variety of types of cells, and the natural fatty acids, which I'll abbreviate as FAs, have an even number of carbon atoms in the chain. And the reason why they have an even number of carbon atoms in the chain is due to the way that they are assembled by the body. The body or the cell, depending upon what type of organism you're looking at, takes 2 carbon building blocks and links those together, and hence the final product has an even number of carbons. The other point that is interesting to mention with regard to fatty acids is that if they are unsaturated, unsaturated means they have a carbon-carbon double bond, and the carbon-carbon double bond, if it is present, will be in the cis configuration. So if they're unsaturated, unsaturated means they have a carbon-carbon double bond. The carbon-carbon double bond is cis due to the way that these molecules are assembled. Now, you may be thinking of trans fats, which a few years ago received some negative publicity for negative health effects. Trans fats are generally not naturally occurring fats. Instead, they arise from industrial processing where cis fats are reacted, and the cis configuration transforms into the trans configuration of the bond. So when we think about these fatty acids, these are the building blocks for a variety of different types of complex lipids, such as steroids. As the building blocks of complex lipids, it is essential that the cell has a way to store fatty acids and transport them around the cell. And the way that the body stores fatty acids is through forming these structures called triglycerides. So triglycerides are how we store fatty acids for later use. So fatty acid Fa, stored for later use. And the way that a triglyceride forms is by the reaction of glycerol. Glycerol is a 3-carbon triol, meaning 3 alcohol groups and 3 carbons. Each of those alcohol groups is bonded to a different carbon atom of the chain here. So we're at 3 carbon atoms and 3 hydroxy groups that are bonded there. And what happens is it comes in and reacts with 3 fatty acids. And I'm going to illustrate one of these fatty acids. Fatty acids have, by definition, a carboxylic acid group and then a long carbon chain. I'm going to illustrate this long carbon chain here as having a grand total of 18 carbons. 18 is an even number, so that makes sense as a naturally occurring fatty acid. And what happens is essentially a Fischer esterification type reaction. Where we esterify by reacting the hydroxy group from the glycerol with our carboxylic acid. Remember that a Fischer esterification reaction is where an alcohol reacts with a carboxylic acid to give an ester product. And so due to the fact that glycerol has 3 alcohol groups, that's why it can react with 3 fatty acids, because each of the 3 fatty acids can link to each of the 3 different alcohol groups to give 3 ester groups present in the triglyceride, hence the tri part of that name. So showing the Fischer esterification product, if we're working just with that one position of our glycerol molecule, we end up with the oxygen from glycerol forming a bond to the carbonyl carbon that originated from our carboxylic acid. And then we have our long carbon chain there, which happens to be 16 CH2 groups, and then our methyl group at the end there to give this esterified product. And if we did that at all 3 positions, we would end up with the triglyceride. So esterify here and here. with 2 additional fatty acid units to give a triglyceride. And there are a variety of different fatty acids within a cell. And so a triglyceride can be composed of 3 identical fatty acids that have linked to the glycerol molecule to give the triglyceride product, or it could be 3 different fatty acids there. For example, a couple of saturated fatty acids, which is what we've drawn here because we have just carbon-carbon single bonds to make it saturated. Or it could be a mixture of saturated fatty acids reacting and unsaturated fatty acids that have 1 or more cis alkene group present in that long carbon chain. Once triglycerides are formed, they are used to store and transport these fatty acids around the cell, taking them to the locations within the body where they can be used to form steroids and undergo other reactions to form the lipids that are necessary for a cell to function or necessary for the body to function. So they are an essential class of biomolecules. We're going to get into this topic a little bit later on in the semester as we look at some types of chemical structures that are related to fatty acids that play roles in allowing us to use chemistry to understand biology. This is the crux of the chemical biology field. So we'll get into that later.